Well, people are still coming in, but I think in the interest of time, I'm going to go ahead and get started. Can you hear me okay in the back? Okay. Well, on behalf of the College of Natural and Agricultural Sciences, CNAS, welcome to the second lecture of the 2015 Science Lecture Series. And the title this year is The Science of Food and Health. I'm Jody Holt, a professor in the Botany Department and Divisional Dean for Agriculture and Natural Resources. So I have the pleasure of working with the speakers and organizers of the series this year. This year we have three sponsors of this series. As I mentioned last time, the College of Natural and Agricultural Sciences, the UC President's Office Global Food Initiative, and then a new institute developing at UCR called CAFE, the California Agriculture and Food Enterprise. And before we start, too, I want to acknowledge our CNAS Science Ambassadors. I don't see but one in here right now, but we have several wearing blue polo shirts. They're standing around in the back, and they're going to be our ushers tonight and assist if you have anything, any questions or anything. And so tonight's lecture is a really special lecture to me because it builds on some work that has been done in the Botany Department for many decades. And it builds on research started by Dr. Anthony Hall, who was at UCR when I first arrived. He's now retired and living in Northern California. And his work on cowpeas made vital contributions to ending famine in Africa. He won the highest award given by the U.S. Department of Agriculture and was quite renowned, and many students came to work with him over the years. Well, the work he started many, many years ago on cowpeas has been continued by the speakers tonight, and I'm really delighted to introduce them to tell you this story of the cowpea, the lowly cowpea, or as Southerners call it, black-eyed peas. So the three speakers tonight are Dr. Phil Roberts, who's a professor and chair of the Department of Nematology, and Dr. Timothy Close, a geneticist, a professor in the Department of Botany and Plant Sciences, and Dr. Jeff Ehlers, who actually formerly worked with Dr. Hall in Botany and Plant Sciences, and a lot of botanists are here to see Jeff tonight, which is nice to see. But he is now a program officer for agricultural development at the Bill and Melinda Gates Foundation in Seattle. And so these three are going to tell us this story, and I'm going to turn the mic over to Dr. Roberts, who's going to tell you about how the lecture is going to go tonight. So welcome, Phil.
Okay, well, good evening, everybody, and thanks, Jody, for a great introduction. There are three speakers, so we thought we'd just give you a little context in terms of how we're going to roll through this. And so we've asked Jeff Ehlers to give up front a broader context of the situation in Africa related to food security, population growth, and this is an area that the Gates Foundation and USAID are heavily funding and supporting. And then following that context, myself and Tim Close will get into some of the details of what we're doing with the cowpea as a crop and modern breeding to advance its productivity for Africa and elsewhere. So that's how we're going to go through, and then we'll have a wrap-up and a question and answer session at the end. So without further ado, Jeff, if you'd like to kick off.
Thank you, Phil, and thanks, Jody, for the nice introduction. So I did want to acknowledge, although Jody's done a good job, the role that UCR has played for this important crop in Africa, being involved for more than 30 years with this crop in developing improved varieties that do well in farmers' fields. And this was mostly funded by the U.S. Agency for International Development, but in 2007 the Gates Foundation also found cowpea and it became one of its focus crops. And when they looked around the world, they said, who can we go to help us work on cowpea? And they came to UCR. And I was proud to start some of that work while I was here. So in terms of the broader context, we'll get down and narrow this down to cowpea. We're faced with a world that we have to feed, and the world is growing rapidly. We don't see that so much in the developed world, and you can see that here since the 1965, for example, in the darker colored part of the bars on the graph. If you look up, the developed world really isn't growing very much, but the developed world, which is in the green bars, is still rapidly expanding and has been since the early 60s. And so we're really looking at going from a world population today of a little over 7 billion to over 9 billion by 2050. So there has been some progress in the world food situation. There's 200 million fewer people now that are considered hungry, and that's given, despite the fact that world population has gone up in that time. So it's a fairly significant increase or decrease in hunger. But still today, 800 million people are chronically undernourished. And again, most of these are in, of course, developing countries. But there's large differences around the world in these developing regions. And while East Asia and Southeast Asia and Latin America have done a pretty good job in the recent decades at reducing hunger, hunger in Africa still is quite severe. And these are the primary geographies that the Gates Foundation focuses in. If we look at the FAO creates food security maps, and you can actually download these at the FAO website. And what it shows is similar to the picture that I described in words previously. And you can see that both India and particularly 3 states, which are Uttar Pradesh, Odisha, and Bihar, not shown on this map, but they're part of the Indian subcontinent. Maybe I can, oops, sorry, I shouldn't have done that. I was going to try to use the mouse as a pointer. Well, maybe that's not going to work. There it is. Okay, I'm pointing to India. So 3 states in India are also our focus countries, and these states also have high degrees of undernutrition. And then the country of Bangladesh in South Asia. So the countries in Africa that we're focused on, you can see there Mali, Burkina Faso, Ghana, and Nigeria in West Africa. And these are countries where actually cowpea is critically important. We also have big hunger problems in Ethiopia, Uganda, and Tanzania, also our focus countries, where cowpea is grown but is not quite as significant. When we look at hunger, though, even within a country, it's not equally distributed. So the wetter areas, which are down toward the central part of the continent, actually don't have near the problems that the drier areas have. So what you see if you look across Africa from north to south is the Sahara Desert, and then increasing rainfall as you get to the center, and then decreasing again as you get toward the tip of the continent. And the band that I've noted there is the area that's right below the Sahara Desert. And with climate change, the climate is getting drier, and the farmers there are really in trouble. And this, paradoxically, is also the region of highest human population growth, and also where Boko Haram and other rebel groups operate. So it's and you can wonder why. It's probably tied to food security. So just to give you an idea of the scale of the problem, I like to show this slide to remind people how big Africa really is. And it's a huge continent. We can see here that all of the U.S. basically fits across the Sahara Desert. All of the Indian subcontinent easily fits into that northeast corner of Africa. Western Europe fits right there in the middle, and even China fits within the whole country. So even though that little sliver that I showed on the previous slide looks fairly small, it's actually quite a huge area where food insecurity is quite bad. So we have today's food insecurity, but then we also have this problem of growing population. And we can see that countries like Nigeria, for example, that have about 175 million people today, by 2050 will almost have doubled their population. And again, this is mostly in the northern region, and again, the northern region is where Boko Haram is. So things are often tied together. We also see in Ethiopia almost a doubling of population projected in the next 35 years. And So we have increasing population, we have current hunger, and we also have declining productivity. The soils are becoming depleted. Fertilizer use in Africa is less than a tenth of what it is in most other countries, particularly nitrogen fertilizer. Nitrogen and water are the primary drivers of productivity, and if you don't have those, you don't have good crops. And because of population growth, then you have continuous farming of the soils and soil depletion, and you don't have the money to put fertilizer on. So you're really kind of stuck in some sense. You can see here a maize crop that's really doing quite poorly, but often mostly due to fertilization, because you can see under the tree where there's been leaf drop and organic matter, you actually have good crop growth. So this is not a water limited crop, it's more of a fertility limited crop in this case. Again, we have the low fertilizer use. Fertilizer is at least 4 times more expensive in Africa, and when you couple that with the low yields they're getting for other reasons, you don't have a lot of incentive to use fertilizer, and it's not really economic in many cases. So what can we do? And this is where we tie in cowpeas and other legumes. Well, one way is to put more legumes into the cropping systems. This slide shows plots that are side by side, and you can see that on the, I guess it would be your right, if I got that right, we have a situation where we have corn has followed corn, and on the left we have corn following a legume, which is climbing beans. And as we know, legumes can take nitrogen out of the air, which is nitrogen 80% of air, can take that nitrogen and fix it and put it into the soil for the next crop to use. So the crop on the left then has benefited from the previous legume crop. And fortunately, there's lots of legumes around the world that we can put into our cropping systems. This is a diagram just showing the sort of phylogenetic relationship among the different legumes, and some are adapted to more cooler areas like lentils and peas that you're familiar with. And at the bottom of the slide shows the warm season legumes, and this includes things like common bean, which is your pinto and kidney beans, and then also things like cowpea and soybean. And the 4 diagrams on the lower left there are different types of cowpeas. You get an idea of the genetic variation within the crop. So cowpea then, among those legumes, is the one that's especially well adapted to the hot and dry zones of West Africa. So it's sort of the crop of choice if you want to improve the soils. And this slide was actually, I took this in 2009 in one of our cooperators' fields at a research station at the national organization called INRA. And they continue to partner with Phil's project and Tim's project today. But it shows you how vigorous the crop can grow with a little bit of good management, and how vigorous it can be in that harsh, hot, dry environment. So Jody alluded to a little bit about what a cowpea is. I developed this slide originally for a class that Tim was holding on food and agriculture, and the question at the time was, what's a black-eyed pea? And I expected the class to kind of chuckle a little bit, and the first thing one of the students raised his hand and he said, "Bad music." So I thought that was a good redundant. But anyway, I guess the black-eyed peas are not as popular as they were a few years ago. But anyway, the cowpea is a black-eyed peas, which many of you know, are a form of cowpea. So it's like black eyes would be like German shepherds are to dogs. They're a type of cowpea. The crop was domesticated in Africa along with millets and sorghum. It's moved around the world, known in Europe in the Roman times, brought to the Americas with the slave trade, where it became part of the food system in the southern U.S. It's actually the main grain legume in northeastern Africa. And just to show the diversity that's available in this species, if you look at the slide at the bottom, you can see that clump of pods or group of pods that fellow is holding, and those are the Chinese long beans, and many of you eat those when you have Chinese food, and that's actually genetically the same as the African cowpea. It's just been selected by man for a very long succulent pod, and you can buy those pretty much in any supermarket. So diversity of kind of uses. So cowpea then is an integral part of the cropping systems across much of Africa. We can see a farmer there in Niger, north of Nigeria, on the margins of the Sahel, growing a crop of millet and cowpea the little green clumps, and he's using a camel to actually pull his cultivator. Quite ingenious. And then we have a Nigerian farmer there with his millet, an intercropping cowpea there with millet. But yields remain far too low, and farmers need improved varieties that resist pests. And Phil and Tim will now showcase the work that they're doing to use modern breeding to help build better cowpeas for the smallholders in Africa. So thank you.
Great, thanks, Jeff. So, we got a big problem on our hands. How are we going to help solve at least a part of it? And so, Jeff just introduced that we have one crop, one plant species, but a lot of variability in it, and we're looking into that variability and utilizing it, and just keying off the long-term breeding program effort here that Tony Hall started. On this plate, they're all cowpeas, okay, but you can see variation in the grain types. At the bottom of the plate are your traditional black-eyed peas, so you're familiar with those. But through breeding, we can breed. On the left is an all-white type that can be used for flour and value-added products. Up on the right side is a green black eye, which you can grow and harvest as a dry grain. But when you rehydrate it, it looks like a fresh-shelled pea, and you can put it in frozen veg packs and so on. So we're talking about a crop here that has tremendous versatility, even within the U.S. And here's some of our seed samples from one of our trials on top left. Cowpeas growing in one of our fields. Most of them here in California are grown up in the Central Valley. And then Jeff talked about the yard-long beans with the very long pods for fresh veg stir-fries and that. So real versatility here locally. And so lessons we've learned here, we've shifted towards our efforts to help deal with some of the African issues in terms of cowpea productivity. So a couple of shots here. Up on the top, there's a forage vendor selling cowpea fodder for animal feed, to feed animals in northern Nigeria. Bottom right is a fairly recently released dry grain type of cowpea in Senegal. Very different to the beans that I just showed you. And then there's further versatility in particularly southern eastern Africa. It's used as a leafy vegetable, so you can make soups and veg dishes. There's a Nigerian market there. It doesn't look much like our Vons or Stater Brothers, but this is where people go to buy their food, those pots of dry grains. And below, here are some young women in Senegal with fresh-shelled or fresh harvested green beans. So a lot of versatility in use. And just to finish off on that, in terms of one of the really popular African fast foods, fried fritters made out of cowpea flour, a wet paste flour. So on many of the urban streets in the cities, in the large cities in Africa, you see these folks preparing food. So really very important. What we've done is targeted work with certain countries. We can't work everywhere, obviously. But we focus primarily in West Africa, working in collaboration with breeders and scientists in their national ag programs. And so using the same map essentially that Jeff showed, you can see along the top zone there, Senegal, Burkina Faso, Nigeria. We're also working in Ghana, just below Burkina Faso. And then in the southern zone, in the southern hemisphere zone, with partners in Mozambique. And so work we do there in terms of breeding in these focus countries often has regional impact because the agroecologies are quite similar, particularly if you're going east-west across these zones. So this partnership is extremely important. It's not easy, obviously, doing work in Riverside and translating that to Africa, but we do have some advantages here. Our climate in Southern California, and particularly in the interior desert area, is a great simulation for growing conditions in Africa. And UCR has a research station out in the Coachella Valley in thermal. And if we plant cowpeas there in August, we can mimic the main growing season in West Africa. So this becomes great for things like drought tolerance screening and other kinds of breeding assessments of our breeding materials. So we have some advantages in this corner of the U.S. So I wanted to touch now and move into some of the kind of the genetic diversity that we're able to utilize. And you've seen this in this photo of the diversity of seed traits, all these different seeds on that photo on the left. They're all cowpeas, all belong to the same species, but tremendous diversity. And when we start looking at resistances to pests and diseases and drought and so on, there is similar diversity within cowpea. There are collections of cowpea germplasm. We have the third largest here at UCR with 6,000 different types of cowpeas. The largest one is in Nigeria at the International Tropical Ag Center, and there are 15,000 cowpeas. So we have a tremendous reservoir of resource for breeding programs. And on this map where we've genetically fingerprinted subsets of these collections, we could identify two main gene pools. The blue dot showing a main clustering, so based on genetic similarity, clustering in West Africa, and then a second gene pool in southern and east and southern Africa, indicated by the red dots, which is thought to be the center of origin for cowpea. So we're dipping into this variation. And trying to advance. And beyond the variation in food qualities and that versatility, one of the reasons we're interested in the genetic variation is that we want to be able to identify and track resistance genes for pests and diseases, and also for drought and heat tolerance. And here I put up a cowpea chamber of horrors, if you will, going along the top from left to right, seedling, flowering, pod filling, and post-harvest stages. And that top bank of photos shows a range of different insect pests that really impact yield. So we have aphids and thrips, bugs that suck on the pods, pod borers, and then post-harvest, the top right, we have weevils that chew on the harvested seed. So lots of impacts in terms of crop production. And then at the bottom, we have a lot of what we call biotic stresses, viruses, fungi, bacterial diseases, nematodes. The bottom right-hand corner is a parasitic weed called striga, which attaches to the root of the cowpea and then grows on the cowpea. So you get a big weed and a small cowpea. So we're interested in blocking these constraints. And that metric at the bottom, the potential, the yield in Africa currently is on average less than 20% of the known potential for yield, the genetic potential for yield under ideal conditions, such as we try to do in California. So there's a huge yield gap here. The good news is that for most of these problems, we have resistant varieties, resistant lines, and I've just shown some pairs here, resistant and susceptible for heat, resistance to heat or tolerance to heat, resistance to aphids, one of our insects, bottom left, resistance to nematodes that attack the root system, and then resistance to this parasitic weed. And so what we do in the breeding program is that we take these sources of resistance and we cross them with susceptible varieties that we want to move that resistance into to improve the variety. And when we do that, we can look at the progeny or the children that come out of that cross and maybe more importantly the grandchildren, and we can look at how the traits segregate within those populations, and then we can track where the genes are in the cowpea genome, which you'll hear more about in a little bit. So here's another example, fungal disease resistance where we have resistance available. So another trait, tolerance to drought. So here's some seedlings that we screen in the greenhouse, cowpea seedlings along the top. You can see they look pretty bad where we've withheld water. So the leaf CNAS going left to right, the stems don't stay green, and then if you put water back on, they don't recover. Compared to at the bottom where there's a line with resistance that has less wilting, the stems are still green, and if you add water back, they will recover and actually recover to give a full crop. So examples of where we have opportunities to move traits into what are currently susceptible varieties in Africa. And in the case of drought tolerance, Africa is mostly rain-fed production. I mean, we grow our crops under irrigation, but if the rains don't come or they come intermittently, then it's a huge problem. So just to lead into Tim's segment, where he's going to talk a bit more on genetic analysis, I'll just take you a little deeper into one example, which is where we're working with resistance to aphids. And many of you are familiar with aphids, those sucking insects that are on your rose bushes at this time of year and many other plants. Well, they're a big problem with cowpeas, both here in the US and also in Africa. And you can see that picture on the left, a cowpea plant smothered with aphids. And they've done a good job of killing another plant there on the right. So an example of one of the approaches is we have cowpea lines with aphid resistance from Africa, like this one on the right, IT97K, a line number, and we've crossed it with one of our California black eye lines, CB27, which is very susceptible, and you can see in the field how damaging the aphids are, and then the resistance holding up on the right. So by crossing those and looking at the progeny, the children and the grandchildren populations out of those, we can identify in the genome where the genes for aphid resistance lie. which chromosome, which part of which chromosome, and then we can track those in the breeding program using markers that Tim's going to explain. And to do that, we need to take leaf tissue to extract DNA to get our DNA fingerprinting. And there are modern techniques where we can run 96 samples through at one time to get multiple fingerprints. So we're using the modern technologies, which are extremely helpful in terms of expediting the effort. And on this chart here, it's one that you wouldn't be that familiar with, but what it does is it indicates in that particular example, we found 2 genes that contribute to the aphid resistance. One of them, the one on the left, is what we call a minor gene, or QTL, quantitative trait locus. We have all these detailed terms, but it contributes about 9% to the resistance. And that's on linkage group or chromosome 1. So here, linkage group 1, this is chromosome 1, 2, 3, and so on. So here's the minor QTL. And then we have a gene that contributes a huge amount of the resistance, 2/3, on chromosome 7. So now we know where those genes are. And now we can go in and track those and move them with DNA markers into our advanced breeding lines. And to allow Tim to key off, here's our sort of a bank of our current knowledge. On these sticks are the 11 chromosomes of Cowpea, and then I've placed on there the names of the resistance traits. So we have drought, heat, all the disease and insect resistances. So we've built a picture of where a lot of these traits are in the genome, and that allows us to then expedite the breeding program. And so with that, I'll call on Tim now to tell you a little bit more on how we do that, and then I'll come back and wrap up. Thank you.
All right, so thanks. So I'll take off from there and give a little preface that one thing that Jeff, Phil, and I all have in common as scientists is we're all geneticists to one extent or another. We have our own various twists on that. But I'll just go ahead and put these phrases up that basically say in words what I'm going to go through with some more illustrations, building off where Phil left off and giving you some illustrations to clarify some of the meaning of these things. So I would say as geneticists, we study the genome of Cowpea, and I'll explain here shortly again what's meant by genome. So what we do is we find positions where favorable forms of genes exist. So here again we're talking about Cowpea has within it a lot of natural diversity. And so there are genes that control this diversity. And there are different forms of them, and we try to figure out where those genes lie. So we do that by using what we call markers, and I'll explain that. We use the markers to sort through offspring in generations after we make controlled crosses where we choose the parents, and we know something about them genetically, and then we follow the progeny. By using markers, we can approach the genotype that we intend to exist much faster than we could without using markers, and I'll illustrate how that works in a semi-technical way. That's the essence of what we call marker-assisted breeding. It's genotype-informed decision-making. So here's an illustration I've chosen that's what's called a karyotype. It shows human chromosomes. So we all, as humans, have 23 pairs of chromosomes, and that constitutes our genome. More specifically, the nuclear part of the genome, that which exists within the nucleus of every cell. The Cowpea has 11 pairs of chromosomes. So every chromosome carries many genes. In humans, roughly 1,000 per chromosome on average. There's then 2 copies of each gene because, as you can see in the illustration, the chromosomes exist in pairs. One is donated by our father and one by the mother. So every chromosome is in pairs. And every chromosome has essentially the same genes along it, so 2 copies of every gene, but the genes generally come in different forms, which we call alleles. So that's that terminology. So markers on a genetic map are quite analogous in many ways to signposts on roads. And so they indicate something in particular at or near a marked location. So here's an example. Here's a yield sign along a road. So the signpost is planted in the ground and it says there's something nearby. It happens to be a lot of different kinds of traffic, bicycle, foot, and horse. And so here's another type of marker. It's saying there will be airplane traffic nearby. And so the signposts themselves can be different. We can distinguish one signpost from another, and they also may mark things that nearby that are different. So there's analogies there to markers that we use in genetics. So here's a depiction of a chromosome, a single chromosome, one of a pair. So we've got a little centromere as a blob and then the 2 arms. So what we do is we develop markers, and I'll explain here again shortly what we mean. We develop markers where we can signpost along the chromosomes, and I've depicted them here in different colors, many with a black head and many with a red head. So we consider 2 different chromosomes that are contributed by our 2 different parents, or with Cowpea we have a different starting point genetically. We'll find that we can mark those same genes, but the markers themselves will be, what I've indicated here is their positions where the gene differs between the 2 forms, red versus green, and then other spots where it's the same gene and it's the same form, and those markers or the genes or markers of the genes are the same when the dark colors indicated. So we can simplify a bit and think about one chromosome in a pair, and we just think about all the red markers and the other just all the green markers, and later on I'm going to come to an illustration where I diagram these essentially as the red chromosome or the green chromosome. I'm talking about differences that we can see within the markers. So the simplest form of marker is one that you can imagine. Here's a nucleotide sequence, so DNA sequence, all the A's, C's, G's, and T's that make the genes. And so the very simplest difference is what we call a single nucleotide difference. So it might be the same sequence but a T at one position and an A in the other. That's a single nucleotide difference, which we call a polymorphism. Okay. So this type of very, very essential polymorphism has been widely used to study many organisms. There's a technology that was developed first for reading these markers in humans. So there's a lot of money that goes into the biomedical industry, and the technology gets pushed forward first for humans. So what we call this genotyping, where you can take a DNA sample, I'll come to this here in the next slide, and extract the DNA and read markers. So I've pulled an illustration here from what's called the Human HapMap project. It's something that started about a decade ago to try to describe all the genetic variation that exists in the human population. And there are now thousands of genetic variations that have been found that are associated with physical characteristics like eye color or disease traits like cystic fibrosis is a good example. So the human genome, all of the nuclear genes, contain about 3 billion bases in total if you add them all up. But it's only about 1 out of 1,000 of those, about 3 million, that differ. The rest are the same from one person to another. So if you want to describe genetically a person, you only need to look at those 3 million places. So there's a technology, it saves money versus looking at the whole genome at every single spot. So there's a technology that's been developed for humans called genotyping, single nucleotide genotyping. And so we've adopted that same genotyping technology for Cowpea. It's possible to do so by engaging with companies that provide these things. So here's a company that was at the forefront of commercializing the human genotyping that was pioneered in the research market. The company's called 23andMe, and they have a little illustration on the bottom how it works. You go online and give your credit card number, authorized $99 charge, and give your address. They'll send you a kit in the mail, and when you get it, you spit into a tube, You collect enough saliva, you shake the tube, it breaks your little cheek cells that are sloughed off. There's some DNA in there. You send it in a padded envelope. They receive it. They extract the DNA. They send you an email that they have your DNA. They run their assay. They look at 600,000 different spots in the genome, and then they post your information online. And up until about not too long ago, a year and a half ago, they revealed a lot of your health information that would tell you if you're a carrier of cystic fibrosis, for example. But there are lawsuits, and at least temporarily, they can only tell you about your genealogy, telling you where your maternal heritage came from and where your paternal heritage came from if you're a male and you carry a Y chromosome. But anyway, an example of what you can see when you can see the health results is illustrated here. This is a screen capture off of their website, of 23andMe website, where if you want to learn a little more about cystic fibrosis, if you're genotyped and you find out, hey, you're carrying an allele for cystic fibrosis, you can dig in and find out what that means. So they have this kind of web portal. So information is not this advanced with Cowpea, where we can read a Cowpea genome and say, this one's aphid resistant, this one's nematode resistant, this one's drought tolerant, but it's moving in that direction, and at least we have signposts that say this area of the genome carries a variant for aphid resistance. We might not know the gene, and we don't need to for breeding, but we know where it is, and we can mark it and follow it. So what we do with Cowpea is we make controlled crosses. So here's an image of a flower the evening before it's open. So you open the keel, and inside are the male and female parts. The male parts carry have the anthers that produce the pollen, and so those you remove with a pair of tweezers. These are pictures of Jeff Ehlers' hands here, and then the next morning you come in and you have an emasculated flower. It's had its male bits removed, and you take a flower that's shedding pollen and dab the pollen. So now you're choosing your male parent by choosing a flower, and you've chosen the night before the female part. And this is the same thing that bees and wasps are doing. They're moving pollen onto stigma. They're making crosses using natural variation. This is what we do in our Cowpea breeding effort, but we're making controlled crosses. We choose the parents, and then we use markers and knowledge of the parents to follow progeny. So I've illustrated here the process that we go through, what we call introgression. Phil talked about aphid resistance, and we've had a successful effort run mainly by Balam Huyn, who's sitting in the back of the room, Dr. Balam Huyn. He's been what we call introgressing, bringing in that African aphid resistance into California varieties. So here's an illustration of that. Here's, let's say, the green genome. Now in Cowpea, like I said, we have a different starting point that we do in humans. Okay, never mind that. In Cowpea, every parent that we start with is completely the same along each of its two partner pair chromosomes. It's different from humans. So the donor here is illustrated as green with aphid resistance as a little yellow star. We know, let's say, we know where that is, and we do. We know where that is in the Cowpea genome by markers and mapping, as Phil described. Here's a current variety, California variety. So the first step is to make a cross, produce a hybrid, what we call an F1, using Gregor Mendel's terminology, first filial generation. So now we've illustrated one complete red chromosome donated from the current variety, and one from this African source that has aphid resistance, but also a lot of traits that we don't want in a current variety. Then you let that plant grow and produce its own pollen, so recombination will take place between the chromosomes indicated by these little X's. You get a lot of different types of gametes out, and I've drawn here the results of 4 examples where you take the pollen from that, you cross it, put the pollen back onto the current variety, so every one of your progeny is going to have a whole chromosome in red donated by the current variety, which we call the recurrent parent, and some recombinational product that came from the F1. So it has part of the green chromosome, not all of it. We use markers to look and see what we have here among the progeny, and we will decide then to go forward with only some portion of them. For example, we decide we want this one. It has the aphid resistance. So then we go through the process again. Take this individual, let it go through recombination. It makes gametes, put its pollen on the recurrent parent, the current variety, and look again, and there will be less of the African variety in the progeny. We sort through again with markers, and here now let's say this is called backcrossing. After the second backcross, we may have a great situation like this where we've accomplished a chromosome that has just one little introgress region with aphid resistance and not much else. So we say we like this one. Of course, we're looking at the rest of all the other chromosomes at the same time and maximizing the amount of the recurrent parent. Ideally, we'd have nothing but the recurrent parent and this one little introgress segment that carries aphid resistance. Then the seeds have to be grown and tested. You have to verify that the plants look the same to the growers as what you started with, and that's in progress. So then you would take an individual like this one, the second backcross, and just let it pollinate itself. So cowpeas have the ability that humans don't. They can self-fertilize and make progeny. So we look for a progeny that looks like this, that has two chromosomes that are the same, and now we've gone to fixation. We have the recurrent variety with aphid resistance. So that's an illustration of introgression. And you can actually take much more complicated approaches and combine things from many different parents by following many different crossing stages and using a lot of markers. Okay, and that's just illustrated here. You can basically now, I'm diagramming here 11 chromosomes. I'm only bothering to draw one each singly, not in pairs. 11 chromosomes. Let's say you have knowledge from 4 different African cultivars, studied them previously, like the map that Phil showed. You know that certain ones carry certain favorable alleles, so you can basically make a painting of what you would like to exist. And now you make your crosses and you use your markers and you follow a path toward building this painting, which we call a genetic idiotype. It's called genetic idiotype breeding. So that's all possible to do, and we have these things in progress. I pulled one example of a recent story from a graduate student who finished recently, a recent UCR example, larger seeds, making larger seeds. So here's a recurrent parent. It's the California black eye variety, and here's a donor, an African variety from Southeast Africa. It has smaller seeds than the California variety, but seed size is controlled by many genes. So this California variety doesn't have every possible favorable allele for larger genes, for larger seeds. This donor, it turns out by previous work, has a gene that makes larger seed that wasn't already present. It's not already present in the California variety, even though the California variety overall has larger seeds than the African variety. So the idea was, go ahead and add this other large seed allele to the California variety and see what happens. And what happened, now you're seeing the rest of the picture, so this is all to scale. This is the resulting line that Mitch Lucas, our PhD student, ended up with. He increased the seed size of the California variety and now made something that's black eyes and larger seed. So that's just one example of the type of things we do that I wanted to illustrate. I'll turn you back over to Phil now.
Well, thanks, Tim. So you're all going to be spitting in those tubes, right, next week? So, it's complicated, and it's really important that we combine different traits. If you have a drought-tolerant cowpea that's aphid susceptible, it'll survive the drought, and the aphids will eat the cowpea, or vice versa. So we're trying to package different traits, and what I wanted to do was just wrap up and give you a sense of product here. And so we do a lot in our fields and greenhouses at UCR. Here are breeding materials for Africa that we're growing at UCR in a greenhouse. And then we work with our collaborators in the African focus countries, the breeders and the African plant scientists, to do collaborative breeding on for their benefit and in their target production zones, and also to work with the agricultural communities to help in terms of seed production, training, and so on. So there's some capacity building and outreach there. But just an example of where we think we're making progress above that lower than 20% yield threshold. Here's an example of some new large seeded white grain varieties that have been released in conjunction with our program in Senegal. And you can see the standard variety, the red bar on the right, Melac. The top set of bars are yield. And you can see that the yields of the new varieties are maybe 20-25% higher in some cases than the standard variety. And the seed size is about 25% increased, as indicated in the bottom graph. And similarly in Burkina Faso, here's a range of new white grain type varieties that are being released, and they have resistance to this nasty parasitic weed and some pathogens. So we're making small incremental steps to deal with the overall problem, and it's a huge problem, but hopefully we've given you a little insight today into how we do that. And here are a few take-home points. We need science applied to this kind of problem. We need sustained international funding. So Gates Foundation, USAID, taxpayer dollars, really important. This is a huge team effort. We have a lot of people at UCR, some in the room and then in Africa to make it happen. And you younger people get interested in science. And everybody in the room, eat your beans, especially black eyes. So thank you very much.
I told you that would be good. Thank you very much. So all three speakers are willing to take questions from the audience. The presentations are being recorded. So if you have a question, raise your hand, and one of our science ambassadors will bring you a microphone. And the person best suited to answer will answer through this microphone, so it's all recorded. Okay?
Yes, no. Oh, yeah.
Thanks. Boy, that was the best tag team talks I've ever seen. Is this on? Can you hear me? Okay. So my question is, I go on the road and I tell people about the fabulous things that are going on here at UCR with cowpeas, but I don't have any metrics. Is there some place where I can go to say, this has contributed to 10% yield increase in Africa, or that so many more people are alive today, or something like that? I know you guys have made tremendous contribution, but numbers are always helpful.
Yeah, so the question is, are there metrics in terms of measuring our progress? And there are some. They're sort of bits and pieces. There have been, within some of the target countries, economic analyses of adoption of new varieties and yields and how that's impacted markets. It's a little bit patchwork. And one of the problems is it takes many years from introduction of a new variety to measure its impact. So there's a strong time lag, but certainly the funding agencies and USA and company are very interested in impact. So yeah, but there are some resources there, so we can get them to you.
Okay, thanks a lot.
The resistance to aphids is what? Is like physiological, is mechanical, is resistant to viruses, or to aphids themselves?
So the basis of aphid resistance, we actually haven't looked too much in the example I gave you of what's happening mechanistically, but well, the bottom line is the aphids start to feed, and then they cannot. They get a bad reaction from the plant sap, and so then they'll stop feeding and die. In some situations, there's more of an avoidance, a sort of an antibiotic approach where they they don't prefer certain leaf types and so on. But we haven't looked in detail at the functional basis of the resistance. Yeah.
Okay. Hi, my name is Delilah. Plant biology major, third year. I had a quick question. I noticed in one of the graphs that Ethiopia was number 2 in terms of the increase in population that's going to come, but it's not one of the countries that you're working with. I was wondering if that had to do because of, is it like environment, or is it just because it's not a food that's used in that area of Africa?
So, yes, I'll take that. So the question was why was Ethiopia not listed as one of the areas that we're working in, or countries that we're working in, despite the large population growth? And actually, excuse me, it is one of our 7 focus countries, but cowpea itself is not critically important there. We do work on common beans and several other crops, but just cowpeas are not emphasized because it's not a critical food source. Thank you.
You mentioned at some point the children and grandchildren, and how long does this cycle take? Is it 10 years?
Yeah, that's a great question. How long are these breeding cycles? And so I'd mentioned analyzing the children and grandchildren generations. Cowpea is a great crop to work with because it will actually go from germination to producing mature seed in as little as 2 months. So we can actually get several generations per year, which is unlike most crops. However, having said that, in terms of starting off trying to develop a new variety, it can be anywhere from 5 to 10 years. The new marker technologies are shortening that time frame, and it depends a little bit on your starting point, but it's typically several years, because once we have the advanced line, we have to field test and make sure that everything else is behaving. Yeah.
What's your favorite way to consume
them? I use a recipe from Selena Ehlers. It's Jeff's wife. It's called Afro-Indian cowpeas. And so I take the dry beans and cook them like beans with a bit of curry and tomato sauce. We can send out the, tomato paste. We can send out the recipe if you'd like.
I want to say that recipe won the UCR academic chili cook-off one year, so I can vouch for it.
So one of the other popular recipes, which I enjoy, is eaten at New Year, which is called Hopping John. And so if you eat, and it has a ham hock in it and onions and seasonings, but it's supposed to bring you good luck. So I don't know how you test that, but anyway, you ought to try it. Yeah.
Question for you, gentlemen. In regards to your research that when you established it in Africa, how easy was that? Were they fairly willing to actually have you guys do work in the area, or was there a lot of resistance to it? I'm curious if there was any difficulty or challenge in actually being able to conduct your research there.
Well, the basis of the relationship starts with the scientists, the collaboration, and then from that, the ability to work with and within with the collaborators and within those countries opens up. And we're kind of joined at the hip because we have collaborative funding. So some of the funding that we get on our major grants goes to the African partners. And so we get wedded pretty strongly. But it's the quality of that partnership that's so important. Even being picked up at the airport and making sure you've got something left when you get to the taxi. Yeah.
I was actually wondering, what are the long-term goals you're actually trying to achieve by that research and genetics and stuff? Because I see that you're trying to increase the crop and make it resistant to the things that kill it, but is that like what you're trying to reach, or is there more?
Okay. Well, certainly one of the goals of the research is to get improved varieties on the ground and improve or enhance the productivity, the amount of cowpeas per unit area of land that are produced. But there are other important things, because we're looking at more basically at the species and the biology of it. We're building new scientific knowledge and understanding, and so that builds a legacy for future generations. So it allows other kinds of research to key off what we're doing. So those are a couple of overarching goals.
So I'll just add, from the Gates Foundation point of view, we're making big investments in the actual delivery of these technologies that are separate from what you see here. So we have, for example, projects that are involving seed production to get the new varieties out. So the UCR folks are mostly on the research end and supporting the research, supporting student training. But then they link with other investments that are actually delivering the seed of the improved varieties, so the farmers can get the genetic gain benefits out of them.
Is it pretty well accepted that the legumes add nitrogen to soil over there? That would be a good crop?
So the question was, what is there evidence that legume crops actually improve the soils, and I guess particularly in Africa? But yes, that's there's no question about it. You can even see it with some of the natural leguminous trees that are in these fields. The farmers don't take these trees down, and these trees are busy fixing nitrogen all year round, and the crops are always super good right under the trees. Because they have a little extra nitrogen. But also many, many years of rotation studies have demonstrated these benefits.
No, I meant, is it well accepted there? Or
Okay.
is there education that has to go on?
So the question, I guess, really was, I misinterpreted, was about is it understood by people in the countries if that there is rotational benefits. Certainly the researchers understand it. There needs to be more work done on extension. There's actually good economic opportunities for the farmers to do the legume production because these crops are worth a lot of money. But at the yield levels they're getting with the varieties they have, the returns are not necessarily as good as they could. So if they can combine high yields, there's definitely markets for them and opportunities, and so they'll be able to reap those benefits.
Is there work or research being done in the countries to see how these new cultivars are being accepted by the people who will be growing them?
Yeah, actually, there's quite a lot of activity in the socioeconomic arena, and and in fact, for variety release, there's quite a lot of testing of consumer preferences and so on. So I talked a lot about white seeded cowpeas, which are really favored at the moment with lots of uses, and so Yeah, I mean, there is a gauging of what that acceptance level is. Otherwise, we'd be kind of shooting off base. So, and there's a lot of farmer participatory selection and sampling, kind of like we do with our avocado program here at UCR. Come in for a taste test. Which ones do you like? So there is that sort of agricultural community involvement that's really important.
I'll just add that what we call farmer preferred variety selection is part of the research protocol. So farmer groups are actually involved in the selection process to help ensure that we do develop things that the farmers actually want and care about. So we understand, through our African partners, what are the traits they care about. Not every variety is a success, obviously, but we have really good data, although we're looking to get much better data based on actually the kind of technology that Tim was talking about, and that is genotyping, because a lot of times once varieties are released in Africa, you lose track of them, they get new names, and so you do have trouble tracking what you've done. But through genotyping, it's basically a paternity test, and you can go back to villages a couple years later and you can actually see, oh yeah, this new variety is still there, it's being multiplied and so on. So we are going to adopt more of the genotyping based methods soon to get better answer.
Okay. Well, I hate to cut this off because there could be a lot more questions, and the speakers will be here for a few more minutes, but I think we should let everybody go. So thank you again, and be sure and come back in 2 weeks for our next lecture.